will be recording. But thank you all for coming. Let's see if anybody else is trickling in here. So, here's very, very good. Couple more people coming in. I am just gonna see if I can make one of you our co-host so that I won't miss somebody who wants to come in. Actually, I don't know if let's see. Maybe I can make Wendy a co-host.
I'm gonna be in and out, so I'm not sure.
Okay.
Sorry.
Just in case you happen to see somebody in the waiting room. And you can let them make a few more co-hosts.
Okay. It'll just pop up an alert and then you let them in.
Yeah. But hopefully I'll be able to pay attention and oh good. We got some more good people coming in here. Excellent. All right. So thank you all for coming. If you are trying to get CE credit for this, CE credit is only given for watching and participating with the live lecture. We will not be posting this lecture publicly. And in order to ensure that you get credit and a certificate, you have to fill out our course survey, and I will post a link to that in the chat towards the end of the lecture. And if you don't see that or don't get it, you can always email me at info@ourhonor.org. And if you have any questions, you can also email me there. And as you watch this lecture, feel free to post any questions in the chat, but we can give people time to answer questions towards the end. And if you want to help us keep these continuing education lectures free and accessible to all, if you are able to donate, please do. We accept Venmo at Our Honor is our Venmo, or you can go to ourhonor.org/donate. And without further ado, I'd like to introduce Megan Barrett, who has a PhD in biology and an MS in education. And she has completed her graduate work in insect neurobiology and physiology, but has used these skills to develop a research program on the welfare of insect livestock. And she has an assistant professor of biology, or she is an assistant professor of biology at Indiana University in Indianapolis, co-PI of the IUI site for the NSF Center for Environmental Sustainability through insect farming, and the director of the Insect Welfare Research Society. Barrett has published hundreds of pages on the welfare of insects as livestock animals, with a particular focus on crickets, yellow mealworms, and black soldier flies. And she has pioneered the first empirically informed standard operating procedures for humane insect slaughter, and in her capacity as IWRS director, helped to create the first guidelines for protecting and promoting insect welfare in research settings. In her spare time, she likes to play with her cats, Nix, Mary, and Pippin, and foster bottle baby kittens with her husband. Thank you, Dr. Barrett, and take it away.
Great, thank you so much. I'm really grateful for all of you taking the time to come to this course today on some of our most misunderstood livestock animals, which would be the insects. So I'm going to talk a little bit about the things on this slide that you can see today. We're going to start actually by just describing what an insect is, because I think sometimes we may not always be on the same page about what an insect actually is. Then I'll move into talking about whether or not we might choose to care about insects by discussing the state of insect sentience research. So I know not everybody chooses which organisms to care about based on sentience, but it is a common standard. After that, I'll introduce you to the insect farming industry and the scale and practices and history of that industry a little bit. And finally, I'll move into some of the welfare concerns that manifest in the farmed insect industry right now for crickets, mealworms, and black soldier flies. After that, we'll have plenty of time for questions and answers. I assume that is something that you will have many of, or I hope it's something you'll have many of by the end of this talk, as there's lots of hopefully brand new material for you on this group of animals. And then at the very end, I have a little bit of group brainstorming that I hope that you'll help me with as an activity to close us out. Okay. So let's start here. What is an insect? So I want to give us a really big kind of evolutionary picture because our insects are not anywhere near the animals that you're probably most familiar with, which is your vertebrate animals. So here we're looking at all the metazoa, which are all of our animals. And this is a phylogenetic tree, which you may be familiar with. So it's just describing the evolutionary relationships between different groups of beings. So here with that red arrow is going to be our chordates. So that's all of our vertebrate animals, all of our traditional farmed livestock are going to fall in there, as well as our fish and aquaculture. All the way over here, that's our arthropods. So that's where our insects are going to be located on this phylogenetic tree. So now let's imagine a couple of other species that you might be interested in, things like earthworms and stuff like that. Those are actually not insects. We're not going to be talking about those today. They actually are farmed and they're used for other applications, but those are annelids. Similarly, we have cephalopods, which are farmed. Those are octopuses, they're mollusks. We're not going to talk about any of those today. We're just going to be talking about one particular group of invertebrates, which is our insects. So if we zoom in on our arthropods, this is what the phylogenetic tree looks like for the arthropods. So here we have a couple of different groups that I want to draw your attention to. The most ancestral group of interest to people is our chelicerates. So that's your scorpions, your horseshoe crabs, your spiders, your sun spiders, your ticks, your mites. All of those are going to be in your chelicerates. So actually none of those are insects either, even though I think many times we think they might be. The next group we have is our myriapods. So that's your centipedes and your millipedes. Actually, those aren't insects either. So another common misconception is that those are going to be insects. They are not. They are other kinds of arthropods. And then we get to this part over here. So that is actually our hexapods or our insects. So all of our insects are going to fall into that part of our arthropods. And that means they're actually most closely related to the crustaceans, the decapod crustaceans like your crabs and things like that, which you can see in blue right here where it says very crustacea. So this whole group forms something called pancrustacea. So insects are crustaceans. It's just something that I think most people don't realize, and they're more closely related to their aquatic decapod cousins than they are, say, to the spiders. And it turns out insects, while arthropods are the majority of invertebrate diversity, insects are going to be the majority of arthropod diversity. And they're also, for that reason, the best studied of the arthropods. So I promise this is the last phylogenetic tree I'm going to make you look at today. But this is us finally narrowing in on just our hexapods. So you can see our outgroup of crustaceans up here all the way at the top. And then all of these are different species of insects that we are going to be looking at today. So basically, to be an insect, what you have to have are 3 body segments and 6 legs. Beyond that, things start to get pretty scary. So the reason they get pretty scary is because insects are super diverse. And so there's not a lot that actually unites them in common when we start to look at all of the species in aggregate, where other than that 6 legs and those 3 body segments, things are really, really different across different parts of the group. So we know already there are at least 1 million described species of insects. We estimate there are 5.5 million total species of insects in the world. And that means there's going to be tremendous variation in things like lifespan, behavior, cognitive abilities, physiology, all of that stuff. Just as one example of that here, all of these photos on this slide are different species of ants. So that's just the morphological diversity you're getting within this one very small group of the insect overall pie. So what I like to typically do now with audiences that may not be super familiar with insects is walk through a couple of axes of diversity that I think are really surprising to people to give you a sense of the magnitude of that diversity for insects, and to perhaps challenge some common assumptions we may hold about insects that make us think they might not be capable of being sentient or having experiences. So one of the first things that I think people tend to think about when they think insects is that they are all really small in body size. And it turns out that is not the case. So insects vary by a factor of about 5.2 million in body mass from the smallest to the largest. You can see a couple of examples of small and large insects on the slide. We've got a featherwing wasp there at the bottom that you can see is really, really tiny. The scale bar on that one is 200 micrometers. But conversely, you can see that that adult man's hand is about the same size as that adult beetle that we have in the upper right-hand side of the screen. So for example, we know that there are some beetle larvae, like the larva of Megasoma aceton, that can weigh 3 to 5 times the mass of a house mouse. So it's definitely possible for insects to get larger than in body mass, even the mammals that we are traditionally familiar with. One other important thing to keep in mind is that we think sentience and feelings and subjective experience evolved, right? It didn't just appear in organisms today when they showed up. It evolved from some ancestor of theirs. And it turns out the insects had some really, really large ancestors in the Cretaceous period. So, for example, the Meganura dragonflies had wingspans up to 2 feet long. I won't bore you with the physiological reasons for why insects we think could get so much bigger back in the day in the Cretaceous period. But suffice it to say, there were some very, very large insects in the evolutionary history of these animals. A kind of related complaint or concern that people have is that, okay, well, maybe insects sometimes get big, but their brains are still always really, really small, right? So let's investigate that claim a little bit further, looking at two kinds of size metrics. We can look at mass, brain mass, and we can look at neuron number, depending on what we're most interested in. So if we consider the smallest mammal brain that has been ever been studied, that's the Etruscan shrew. It weighs about 64.4 milligrams. The largest insect brain we've studied to date weighs 11.7 milligrams. And that is not by far the largest wasp. So that wasp that brain came out of is a 0.55 gram wasp, but we know that insects get up to 200 grams in body mass. So we can expect much, much larger brains than that particular wasp's brain. As far as vertebrates generally, if we're willing to consider our reptiles as plausibly sentient, it turns out our smallest vertebrate brain that we know of is 11.3 milligrams, so actually smaller than the brain mass of the wasp. That's from the Algerian sand gecko. Now, if we look at neuron numbers, it turns out that the story is really similar. When we consider the Algerian sand gecko and the largest insect, we see that they both have 1.76 to 1.77 million neurons, so basically exactly comparable. When we look at the smallest mammal, we see that it has 27 million neurons. That's your naked mole rat. So that's only about 15x bigger than, again, the wasp, which is not even the largest insect we could study. And interestingly, that means there's more variation in the mammals than there actually is between the largest studied insect and the smallest mammal. Because, for example, if we look between the naked mole rat and the elephant, we see there's a 1000x difference in their neuron numbers compared to the only 15x difference between that wasp and the naked mole rat. So all of this is just to say that insect brains plausibly aren't too small to be able to do a lot of things that vertebrate brains do, because in some cases they're actually size comparable. Let's think about life history. So people tend to think of insects as kind of like the cheap model organism. So they produce a ton of offspring, they don't invest anything in them parentally, they live really short lives, they reproduce quickly, they develop quickly. All of those things can be contested depending on the species that you're talking about. So when we think about reproduction, there's actually both the feature of laying many, many eggs that you're probably familiar with for insects, but there are also insects that actually birth live young. So viviparity, which is, developing live young and laying live young, that has actually evolved 11 different times across the insects. And there's one fly species I find really compelling, where the female actually has kind of she has a uterus inside with a milk gland, and she feeds her larvae off of that milk gland in utero, develops them one at a time, giving birth to them right before pupation. Those females average only 4.5 offspring over the course of their entire life. So clearly they do not produce small numbers of offspring with very little parental investment. As far as parental strategies, of course, we have things like that fly I just mentioned. We also have monogamous biparental care, for example, in termite colonies where queens and kings can live together for 20 years, taking care of their offspring in the colony. We have solitary individuals of either sex that can provide care for their offspring, like you're seeing at the bottom of this slide. We have earwigs and hemipterans and water bugs, each providing parental care to eggs or juveniles' offspring. And we have tons of variation in lifespan too, which I think is something that people often forget. So we have juveniles that can live for up to 17 years in the juvenile life stage. We have adults that can live as little as 5 minutes in the mayflies, or even up to 28 years, like queen ants of some species have been recorded to live. So you can imagine that if you're living for 28 years, you might have a lot more need for learning and the kinds of adaptive value that we think sentience can provide compared to something that may live for, just 5 minutes. Lastly, I think people tend to think of insects as cognitively unsophisticated and behaviorally really simple. And this is also contestable. The more we learn about insect behaviors, the more we realize they can be quite cognitively sophisticated and their behaviors can be very flexible and complex. So I've included just a couple of examples of behaviors here from across the insect group. But a couple that I'll call attention to really quickly. We have rescue and wound treatment behaviors in ants, where when they get into fights with termite colonies, they'll actually carry their injured nestmates back to the nest, tend to their wounds with antimicrobial secretions, and that will increase the survival probability of those injured nestmates by about 90%. We have individual facial recognition in wasps, and they can actually do transitive inference as well. Like if, wasp A that I can recognize as individual A has beaten up wasp B, and I know wasp B can beat up me, then I'm not going to challenge wasp A. We have emotion-like states, and this includes in studies of fruit flies that are used for biomedical research on things like chronic pain, anxiety, and depression states. So you can actually treat flies so you can do forced swim tests with flies like you can do with rodents. You can treat them with antidepressants, and it will change their performance on those tests. So I guess if there was a takeaway of this insect diversity section, hopefully you're a little more familiar now with just how diverse they are and some features of their nervous systems and their body size and their life history and their behaviors. But I'm hoping that you'll be suspicious of your intuitions. That's because we should probably all, myself included, be suspicious of our intuitions about insects, because assumptions are really common, but they're often incorrect about this group. The most readily available information is often not correct, especially online. There's a lot of misinformation about this group of animals. So it's really important to check with experts and disciplinary specific experts. So if you're interested in insect pain, check in with researchers who don't just research insects, they research insect pain on the neurobiology or behavior level. And this is especially true, I think, for veterinarians when you're considering insect versus vertebrate physiology, because there are many really important differences like metabolic strategy, ectothermy versus endothermy, that are going to be important and bear on the welfare concerns of these animals. So it's important we keep that in mind as we're starting to think about how we might improve their welfare in farmed conditions. Okay, so now let's move on to this question of whether or not we think we might want to care about insects at all. Are they sentient? Could they plausibly feel pain? Could they plausibly suffer? So the standard citation you're likely to get on this topic if you contact most entomologists right now is this 40-year-old paper, Eisman et al. 1984, Can Insects Feel Pain? A Biological View. And in it, they write that insects have no nociceptors, which are the sensory cells in our peripheral nervous system that are responsible for causing us to respond to noxious stimuli. They have a very simple nervous system organization, so they posit that more complexity in the nervous system is necessary to produce the experience of pain. They don't say specifically what kind of complexity is necessary, but more recent authors have addressed that. So for example, Shelley Adamo in 2019 wrote that it's the lack of direct connections between the sensory integrative brain regions that makes it unlikely insects can feel pain. And they also cite some behavioral evidence. So they see ants walking on crushed limbs. And although they do see some site-specific grooming in response to injury sites in cockroaches, that grooming didn't end up being adaptive, so it didn't improve survival for the organism. And of course, we think pain is expensive, so it probably has some adaptive value. And so if there's no adaptive value, then that probably isn't a pain-relevant behavior in Eisman's view. So altogether, this paper comes to the conclusion that there is likely no adaptive role for pain perception in insects. So the problem with this paper, and with the fact that people keep citing it, is that it is 40 years old. And as you might suspect, given that you were in a continuing education course, things change and you have to continue educating yourself on the best available evidence. And the evidence has changed a lot in the last 40 years. So let's revisit a couple of these claims in the 21st century. First, we now know that insects do have nociceptors. They've been discovered in both larval and adult insects. They have nociceptive ion channels that are homologous to ours. Those are the proteins embedded in the membrane of the nociceptive cell that are going to kind of cause the cell to fire in response to the noxious stimuli. So they have TRP channels that are homologous to our TRP channels for things like heat and stuff like that. In terms of the simple nervous system organization, we now know their nervous systems are much more complex than we realized in the 80s, and even actually more complex than Adamo realized in 2019. The very next year, we did the first complete map of all of the connections of the adult Drosophila melanogaster brain, and we found direct connections between the mushroom bodies and the central complex, which are the two sensory integrative brain regions she calls out in that 2019 publication. As far as the behavioral evidence, you're probably already aware of why some of that is not compelling evidence against pain, but it bears repeating. Obviously, many sentient beings behave as though they're not injured when they are injured. Prey animals, for example, not limping on broken legs like cats and things like that. So we have to be careful interpreting behavioral evidence in the negative sense. And as far as the site-specific grooming, like I mentioned to you earlier, there have now been several studies on ants in wild settings that demonstrate that when you're not in a sterile lab, that grooming is actually adaptive in those conditions. So this is not to say insects feel pain and are sentient. It's to say if anybody ever gives you the Eisman paper, you should tell them it's a bad citation and not to cite it anymore because it's 40 years old. We need to move on, right? One way we might move on is to ask the question, what pieces of evidence might matter to us in thinking about whether a group of organisms is likely to be sentient or feel pain? Once we've established those criteria, then we can go through and look and see how many of those criteria a particular group of animals fulfills. So this is what Jonathan Birch's group in the London School of Economics decided to do in 2021. They developed this framework for invertebrate sentience, which they then applied to cephalopods and decapods. That's your octopuses and your crustaceans, and like your crabs and lobsters and things like that. And so this framework is based on the Smith and Boyd framework from 1991 that was developed for vertebrates. And it's just a slightly more inclusive and functionally focused version of that framework. So for example, that framework specifies the animal has to be affected by opiates specifically. Many invertebrates have other kinds of endogenous analgesics. So now this just says endogenous analgesics instead of opiates specifically. That's how it's focused on function instead of focused on specific chemicals. So this framework has 4 pieces of neurobiological evidence and 4 pieces of behavioral evidence. And the idea is that the more of those pieces of evidence a group of animals meets or is able to fulfill those criterion, the more likely it is that they are going to be potentially sentient and capable of feeling pain. And so we should be morally cautious in our treatment of them. I want to be really clear that this framework is not providing a probability of sentience estimate. So it's not like if you meet 4 of 8, you're 50% likely to be sentient, right? It's just accumulating evidence to help us decide when to act cautiously. And so it's also not like if you meet 8 of 8, you are 100% for sure sentient. It just means we have a strong and compelling reason to act carefully towards that organism until we do know whether or not they are sentient with some definitive bar. Okay. So this particular framework, the reason I'm going to call our attention to it today is because it has actually been decision relevant in some contexts for protecting invertebrate animals. So when we consider cephalopods and decapods, which is what this framework was originally developed for, the octopuses are going to meet 7 of 8 criteria to a high or very high degree of confidence, which this framework rates as very strong evidence for sentience. The decapods meet 5 of 8 to a high or very high degree of confidence, which is strong evidence for sentience. On the basis of this report and this evidence, the UK government actually chose to include both of these groups in their animal sentience bill in 2022. So this was enough evidence for the UK government to say some caution is required and we should be more considerate about these animals. So 5 of 8 is kind of our bare minimum standard at this point for maybe choosing to be cautious. So where do insects stack up? With the group of authors that did that original publication on cephalopods and decapods, as well as some other insect biologists, led by Matilda Gibbons, who's a fantastic postdoc at University of Pennsylvania now in the United States. We looked at 6 different orders of insects at the adult life stage and at the juvenile life stage separately. So you're seeing those 6 orders listed out on the left-hand side of the screen. We have Blatodia, that's your cockroaches and termites. We have Coleoptera, that's your beetles. We have Diptera, that's your flies and mosquitoes. Hymenoptera, which is your bees, your ants, your wasps, and your sawflies. Lepidoptera, which is your butterflies and moths. Orthoptera, which is your crickets and your grasshoppers and katydids. We chose those 6 because there was the most evidence on those 6. There are many other orders of insects. They just aren't very well studied. And so we weren't able to accumulate enough evidence for it to feel worthwhile to add them to the publication. So I think there's a couple of key results to kind of pull out from this table. The first are these 2 red highlights. So you can see that for the Blatodia and the Diptera, so cockroaches, termites, flies, and mosquitoes, they meet 6 of 8 criteria to a high or very high degree of confidence. So this is 1 more than the decapod crustaceans like crabs, 1 fewer than the cephalopods like the octopuses. And so already this suggests to us that we have some strong evidence for sentience in those groups. Now, the reason those groups jump out at us most likely is not actually because they are more likely to be sentient than the others, but rather because they both contain model species for the study of nociceptive neurobiology or pain research. So the fruit fly and the Diptera is a really big biomedical research organism. People study chronic pain in that animal. So for that reason, they end up, fulfilling a lot of these criterion because we've investigated their nociception and pain capacities a lot. When we look at the Blatodia, the American cockroach has been a study system for nociceptive circuitry for a long time as well. And so it meets many of the behavioral and nociceptive criteria listed here. So it's not necessarily that they're more likely to be sentient, it's just that they're better studied in this particular regard. One other result that I think could be interesting to point out to people is that we only found 1 piece of evidence that pointed against the capacity for a group of animals to potentially fulfill a criteria, and that was for Hymenoptera for criterion 8. So criterion 8 is analgesia preference. This is the idea that an animal that has been injured will prefer either a location where it's receiving an analgesic or a food source that comes with an analgesic, something like that. So in this study, Groening et al. 2017, they clipped the legs of honeybees while under anesthesia, allowed them to wake up, and then gave them access simultaneously to a sucrose feeder, then a feeder that had sucrose and morphine in it at the same time, and quantified whether or not the bees drank more from the morphine-laced feeder than the regular sucrose feeder. They did not. And so they concluded that this meant that the bees were not experiencing analgesia preference in this case. There's a couple of challenges with saying that this study provides strong evidence against the capacity for insects to fulfill criterion 8. One is that, as you may have caught earlier when I was talking about the framework being more inclusive for invertebrates, and one of the ways that it became more inclusive is by getting rid of the opiate-specific function. Insects do not have opioid receptors. So we have genetic data that demonstrates there are no opioid receptors in insects. That means that opiates are not currently considered a validated analgesic for insects. We do actually know they appear to have analgesic-like functions that could be based on off-target binding, but we have no mechanistic understanding of why those analgesics might be having analgesic-like effects. And until we do, instead, it would be better to use analgesics that we actually do understand the mechanisms, like GABA agonists and things like that. The second problem with this would be, if you're familiar with this kind of experimental design, typically you don't actually give the animal simultaneous access to the analgesic and the non-analgesic treatment without any preconditioning. And that's because the morphine's impacts, if it has any, are not going to occur immediately. So the bee could walk up to the morphine-laced sucrose feeder, start drinking from it, move over to the other feeder, start drinking from that one, and feel the effects of morphine as it's drinking from the non-morphine laced feeder. That would, of course, cause it to associate the effects of morphine with the wrong feeder and ruin your ability to get successful results. The final problem with this is that we do know that morphines have taste receptors that are that bees have taste receptors that are sensitive to morphine, and it tastes bitter to them. So they actually dislike it. They find it aversive. So now you're asking them if they like an aversive feeder more than a non-aversive feeder after having been injured and being potentially in pain. Overall, not providing strong evidence against the capacity for insects to fulfill criterion 8, though of course more evidence is required. The other thing I like to do is put the evidence that we're acquiring back into a phylogenetic context, because again, sentience did not just appear in cockroaches and appear in fruit flies. It evolved presumably from some common ancestor. So if these groups are really closely related to each other, then we might expect just that group of insects to have these capacities, where if these groups are pretty far apart in where they're related to each other, it's probably most likely a common ancestor of both of them develop these capacities. And so this might be a more widespread character across the insects. So what we're seeing here is the red arrows pointing to the Blatodia and the Diptera, which are two groups that met 6 of 8 criterion at the adult life stage. The blue arrow is pointing to the Hymenoptera that met 4 of 8 criteria at the adult life stage. What you can see is that those red arrows are distributed across that thick black bar that I've put in the middle of the screen. So that black bar is separating a major kind of division in the evolutionary history of insects between the holometabolous insects and the hemimetabolous insects. So the holometabolous insects are those that undergo complete metamorphosis. So here you can think about flies and bees, butterflies and beetles. They start out like some little grubby thing, some little maggot or worm type thing, and then they pupate. They're in the chrysalis, they're pupating, they turn into goo, and they emerge as some radically different looking animal, as a fly, as a butterfly, as a beetle that looks nothing like their larval stage. That's holometabolous development. We also have hemimetabolous development, which is incomplete metamorphosis. This is actually the ancestral strategy for insects. So it's what the oldest insects tend to do, even though only about 20% of them are actually hemimetabolous at this point. So incomplete metamorphosis is when you start out, you emerge from the egg looking like a little tiny version of what you're going to look like as an adult, minus like your genitalia and your wings. And then you just become a slightly bigger and bigger and bigger version of that until you molt into being an adult. So crickets are a phenomenal example of that. They start out as little tiny crickets and become successively bigger crickets until they get their genitalia and wings and they're adults. So the interesting thing here is that these red arrows are distributed across that divide. And this suggests that a lot of the characters that these two groups are meeting that are providing some evidence for sentience are actually likely to be quite ancestral in the insects overall. We also did look at larvae, more, I should say, at the juvenile stage, because some of these are not larvae at the juvenile stage, like the crickets and the cockroaches. And I think the main story to take away from our work on the larvae or on the juveniles is just that people don't work much on juveniles. There was not a lot of literature to read. This makes sense as somebody who's tried to work on juvenile insects some in the past. They're much trickier to work on than adults because they're constantly molting. And that presents a lot of challenges to your work. That said, I guess there's one result still to highlight, which is the diptera again. So the most studied juvenile insect is the larval fruit fly. They meet 4 of 8 criteria to a high or very high degree of sentience at the last instar, which is when they would be slaughter age if they were being farmed as a comparable dipteran species. And they're very likely to meet 5 of 8 at the last instar, that one medium for criterion 3, which is integrated nociception. We're just missing one link between in like the whole integrated nociceptive circuit, but it is highly likely that that link is going to be found based on the behavior and capacities of the animal. I also want to draw your attention to 2 studies of what I think are like special significance in making me think harder about the plausibility of insect pain. The first is the study by Kwong et al. In 2019 about nerve injury driving a heightened state of vigilance and neuropathic sensitization in Drosophila adults. So that's fruit fly adults. They basically give a peripheral nerve injury to the legs of these fruit flies, then determine that many, many, many days after that injury has been given, those flies are actually much more sensitive to temperature than they had been before. So temperatures that previously were mild and non-aversive have become aversive to those animals after receiving that peripheral nervous system injury. The other study came out this year, and it's about Drosophila pain sensitization and modulation being unveiled by this novel pain model and analgesic drugs. So in this study, they provided, first they genetically modified Drosophila adults to be expressing a human TRP channel in their mouth parts. So remember those TRP channels I told you about before are the ones that open and cause cells to fire in response to a noxious stimulus. So they've taken that protein from humans so that we know its function, and they've put it into the mouth part cells of the flies. That protein is there, and this particular protein fires in response to capsaicin. So flies do not typically respond at all to capsaicin because they do not have these TRP channels. But now they have these TRP channels, so they should respond like people do to capsaicin, which is to say there should be some indication of potentially pain. So what they found is that if they provided capsaicin-laden foods to this genetically modified version of Drosophila, they would start to eat, immediately stop eating, start to pull at their mouth parts and run around frantically trying to clean their mouth parts off. And then depending on the dose of capsaicin in the food, there was a dose-dependent lethality effect where the insects would actually starve themselves to death over choosing to eat the capsaicin-laden food if the dosage was high enough. So at small doses, they were willing to kind of go and eat a little bit of it. But at high enough doses, it was potentially noxious enough to them to cause them to actually starve to death over choosing to consume it. They then tried giving those same modified Drosophila some common analgesic compounds, so NSAIDs, opiates, GABA agonists, and others. And they gave them things that are not expected to be analgesics and saw whether that rescued their survival in response to the extremely high doses of capsaicin in the food, and found that it did. So basically, they're proposing that this particular model system can be used to screen for analgesic compounds, where if all your Drosophila are dying, it suggests that this is not a good analgesic, whereas if you rescue the survival phenotype, then this is a good indication that the compound that you're giving these animals is analgesic and pain relieving to them. These 2 studies suggest that there are more conserved mechanisms for pain relief and pain experience and things like neuropathic sensitization between mammals and invertebrates than I had previously suspected there would be. So to me, they're much more compelling pieces of evidence, even than some of the things I just reviewed on the preceding slides. So if I have some main takeaways for you on the do we care about insects, should we care about insects, do they feel pain part of this talk, hopefully there's more evidence of insect sentience and pain than you may have expected coming into this talk. Evidence is distributed across that major divide in evolutionary biology, that holo and hemimetabolist divide. But as I mentioned to you, there are a lot of key research gaps, and that's especially true for juveniles. But I think potentially another main takeaway here is just that there are these decision relevant frameworks like the Birch framework that we see stakeholders that we care about, like governments or producers, that they're using these to decide when they want to protect beings before we know whether or not they're likely to be sentient. And using that same framework and applying it in a consistent way suggests that at least one highly farmed insect species, the black soldier fly, should be protected out of caution. Okay, so now we are actually going to move into the part of the talk where we talk about insect farming, which is an industry that is very novel in some ways, and so you might not be super familiar with it. So the insects as food and feed industry is basically brand new at the scale that it's currently operating. So people have been wild harvesting, eating insects for millennia, and people have had small scale farms for a long time, either to produce exotic pet feed or zoo animal feed, or even for human consumption, especially in some Southeast Asian countries, and African countries and Latin American countries. But in the West, rearing them at scale is very new. Insects are a protein source. You can consume them if you're a person, depending on the species, or farmed vertebrate animals can consume them, again, depending on the species. There are about 2,000 species of edible insects that we know of so far. And the main selling point of the insects as food and feed industry is that it can be a more sustainable source of protein than vertebrate farming is. There are a lot of caveats about this that I'm not going to get into today because the point of today's talk is not a sustainability analysis. Basically, what I'll say is that generally you get more sustainable results out of this industry if the insects are eaten directly by humans in place of vertebrate animals, and or if they're fed very specific waste feeds that would otherwise not be utilized and put back into the food system. Importantly, they could be used to improve food security in protein-poor areas where there's not very much vertebrate agriculture and there's a lot of kind of climate change-based agricultural insecurity. And because this is a global industry and I don't have time to introduce you to all of the variation in it today, just know there's a tremendous amount of variation in the practices being employed. You have everything from highly automated, practically entirely robotic, gigantic farms in the West through little modular units that are being employed in the UK directly on vegetable farms or poultry farms, all the way to, like rural concrete pens that people are rearing crickets in their backyard in Thailand. So when we say insect farming, it covers a wide variety of practices, but mostly today I'm going to be talking about the mass-produced level, so that highly automated kind of Western industrial practice. So this is the current scale of the insects as food and feed industry, looking at the main taxonomic groups that are farmed. As of 2020, we're seeing about 1 to 1.2 trillion estimated insects farmed each year. And I do want to note this is, again, as of 2020, which is a couple of years ago at this point. I want to put that scale in perspective for you. So if we're looking at billions of individual animals on this chart on the left-hand side, all of our terrestrial vertebrates, so chickens, pigs, cattle, all of those are in that little green bar. So there's about 80 billion terrestrial vertebrates slaughtered each year. For insects, again, our mean is 1.1 trillion as of 2020. So already we are farming many, many, many more individual insects than we are vertebrates. And that's just considering the insects as food and feed industry, which is what I'm going to talk about most today. But there are also other sources of insect farming that we should probably be aware of. And if we added these to our calculations, the numbers would surely get even bigger. So you may be aware of some of these species control applications from your work with vertebrate livestock, where, for example, screwworms are a really big parasite problem for many vertebrate livestock. So we have used sterile screwworm breeding and release programs as a way to control that species and reduce impacts on livestock animals. That is not the only species control application. We rear many species of insects for species control. We obviously produce silk from silkworms. We produce dyes from cochineal insects. We produce shellac from lap bugs. There are lots of bees that are reared for various pollination services, and we're even seeing a novel use of fruit flies as growth medium for producing cell-cultured meat products. So why am I focused on insects farmed as food and feed today? For two reasons. One, it's because I can't be an expert in all of those things, and so I primarily am an expert in this one, so that's what I can share with you the most. And it's also really relevant from a livestock perspective. We often tend to focus most on those that are produced for consumption. But also it's because of the growth of this industry. So even though many of these other industries actually do rear a lot of insects, like what you're seeing here for silkworms in the silk industry, we rear almost 2/3 as many just silkworms as all of the insects as food and feed in 2020. But the silkworm industry is pretty stable, so it's not growing very rapidly right now. And that is not the case for insects as food and feed. This is a super rapidly growing industry with significant investment capital, like what you're seeing here. So if we're looking from 2015 to 2020, we can see that in 2015, there's maybe about 25 million US dollars flowing to insect farming companies. By 2020, we're looking at over 430 million US dollars that year flowing to those insect farming companies. And that is because rapid growth is expected in this sector. So this is a graphic produced by the World Wildlife Fund and Tesco in 2021 about scaling up the use of insects in feed in the UK for poultry and pig production. So they are expecting a 10-fold increase in production with a 22-fold increase in demand by the end of this decade. So even if they are able to meet that tenfold increase in production, they will not meet the increase in demand for insect protein, is their estimation. The reason for this tremendous increase in demand is because of this expansion into novel markets. So most of the old market for insects as food and feed was novelty food products, like a very small novelty food product segment, and then exotic pet feed and zoo animal feed. But the largest market that is expected to grow in the future is fish meal replacement for aquaculture. So the UN has actually estimated that we could replace up to 100% of fish meal sustainably with insect protein instead. If we are a little more conservative about that, and we estimate we replace just 25% of fish meal with insect protein, that's an additional 40 to 80 trillion insect deaths each year. So that's what that looks like on a graph. It's the lower of those two red bars. Our orange arrow is pointing to our 2020 levels of farming for insects as food and feed. So we can tell vertebrates are now completely missing from the graphic because their numbers are just so small. The upper of those two red bars is the worst kind of case scenario, if you will, from potentially a welfare perspective, where we'd be seeing 100% replacement of fish meal with insect protein in that particular industry. And it's important to keep in mind that when we look at that 25% and 100%, that's just considering fish meal replacement. It's not considering any replacement of pig and poultry feed, or inclusion in ruminant feed or cattle feed. It's not considering exotic pet food, human consumption markets. That is just considering fish meal replacement. So the potential for the industry to grow is really, really large. The potential for sustainability benefits is very large. And the potential for animal welfare harms, if these animals can feel pain and there are welfare challenges, is also very, very large, and we should take it seriously. So let's go back to considering our main farm species. We've got crickets, yellow mealworms, and black soldier flies. I'm going to give you just like super fast facts about these different groups of animals because they are different species. So we should treat them as differently as chickens, cows, and pigs. We should not think of them as insects. We should think of them as individual species. So for the black soldier fly, this is a fly. It's in the Diptera. It is holometabolous, that means. So again, it's the kind of insect that has a larval stage, pupates, goes through complete metamorphosis, and then becomes a radically different adult form. So these reach pupation in about 14 to 21 days, although most of them are actually going to be slaughtered to become product at the larval stage. So most of them do not actually pupate. The ones that do are pupating because they become adults to become breeders for the colony, and adults can live up to 40 to 70 days. One of the main attractions to this species for the industry is that the larvae can eat really weird stuff. So this is a filth-feeding fly by nature. You find them in manure and compost and other kinds of degrading substances. As a result, they have a really highly, they have a great immune system that has evolved to live in those kinds of microbially rich substrates. They have a lot of antimicrobial peptides, and they can survive a lot of potential diseases and microbially rich environments. So this means you can utilize a lot of waste feeds that right now are just going to waste instead of being used to be put back into the food system and turned into protein. Now, these larvae do live buried in the feed. So you provide them with some food, they bury into it, and that's because they're photophobic. So they are afraid of light. When they become adults, their mating strategy in the wild is that adult flies will leave where they have pupated and then emerged as adults, fly to some trees. The adult males will fly to trees and leck there. So lecking just means they aggregate and then they stake out little territories and wait for females to come in, find them, mate with them, and then leave. So females emerge, they find the lek, they mate with a male, they fly away from the lek to a site to lay eggs, and then they'll lay these large clutches of eggs there, and they could potentially return to the lek a second time to mate again. Our next kind of focal species is the yellow mealworm. So this is a beetle species, Tenebrio mollitar in the Coleoptera. It's also holometabolous, so larval stage, and then turns into an adult post-pupation. Again, mostly slaughtered at the larval stage for this species. So the only ones that are allowed to reach adulthood are the ones that become breeders for the population. These ones have a much longer life cycle. So they typically reach pupation in 60 to 90 days, but depending on how you're rearing them, it actually can take as long as 2 years for them to reach pupation. Adults can live between 1 and 6 months breeding and laying eggs in the substrate. Larvae typically eat grains. So these are actually stored product pests originally. You find them in flour stores and things like that. They may be able to consume mycotoxins and plastics safely, which is another reason why people in the industry are interested in them. Mycotoxins obviously contaminate grains. They're produced by fungi, and they make it incompatible for humans or vertebrate livestock to be able to consume those grains, but it's possible you could feed those contaminated grains to yellow mealworms and then feed the yellow mealworms to the vertebrates. This species has a lot of cannibalism, and this is part of its natural history, actually. So the pupal stage, typically insect pupa, if you imagine a butterfly chrysalis, it's just a tube. And the pupa is kind of goo in there doing its, morphological revision. They have actually evolved in the case of Tenebrio molitor. The pupa has a tail flicking ability, and that flicking is thought to be an anti-cannibalism behavior, where if it gets touched by anything, it will flick at it to try and get it away so that it doesn't get eaten by its species mates that are living in the substrate with it. They also live buried in their feed, and they are also photophobic. Cannibalism can happen at both the adult and the larval stage. In particular, cannibalism can also be common when the insects are molting or have just emerged as adults, because their cuticle, their exterior skin layer, is very soft then. It takes some time to harden. And so before that hardening happens, they're very vulnerable to being eaten. So when you're rearing them in high density conditions, they are more likely to be encountered when they're in that vulnerable stage and more likely to be consumed by their conspecifics. Adults also live and mate in the feed. They're also photophobic. They have tonic immobility responses to predation threats, where they will pause and wait in response to those kinds of predation stressors. The last group we're going to talk about is the crickets, and we're actually talking about a kind of group of 4 different species here: Echinodomesticus, Gryllus assimilis, Gryllus bimaculatus, and Gryllodes sigillatus. So the industry mostly reared Domesticus, and then there was some really, really big disease problems. This Denza virus came through the population and caused a ton of colony collapse in Western Europe and America. And so they switched to some more resilient species, like the Gryllus species and the Gryllodes species, that have proven to be a bit more resilient to Denza virus. So these are hemimetabolous species. So that means that they start out as little tiny crickets and then just become bigger and bigger ones. They're typically slaughtered at the early adult stage. So most of them will be able to reach adulthood. Their lifespan is about 90 to 120 days total. Some of them obviously survive longer in adulthood to lay eggs and things like that for the colony. As far as their natural diet, they tend to be scavenger species. So they eat vegetation, they eat dead and dying insects they come across, and other random protein sources they might find. They tend to also be photophobic, and they tend to be extremely sensitive to predation stress, which they sense as vibrations through the substrate that they are living in or the ground that they're standing on. So it can be good to be aware of that part of their biology as well. In these species, adult males tend to be highly territorial and very aggressive. So they will fight with each other for access to mates. This can lead to some serious injuries sometimes. And this also is a species, or they are a set of species, that can exhibit cannibalism. In particular, A. domesticus has been known to exhibit a decent amount of cannibalism, which they call self-thinning. So if you rear them at too high of a density, they will thin their own population out by eating one another. There is another mechanism available to some of these species in the wild for reducing the number of individuals in one area at a time, which is that they have density-dependent wing development. So juveniles, when they're molting into becoming adults, if there are a lot of other juveniles around them, they will develop longer wings in some species so that they can fly very far away and disperse. If there are not that many individuals around them, they will develop short wings. They will stay in the local area and begin to mate and reproduce immediately. So this density-dependent wing morphology feature is meant to aid in dispersal when densities are too high in your local area. Obviously, on farms, they can develop those long wings, but there's nowhere to disperse to, and so they might resort to other tactics for self-thinning. So probably from the slides that I've already discussed, you can imagine that there are some welfare challenges already in those slides mentioned for farmed insects, but I'm going to go a little bit more in depth now on that. So I'm going to do kind of a case study on the black soldier fly, and I'm picking that species because it is expected to become the most farmed species moving forward. So it's not currently the most farmed, but it should be in the next decade. So black soldier fly farming looks something like this. You find some waste source and then you process it so that it's safe for the larva to consume. That might mean removing some water from it, adding water to it, changing the macronutrient profile, etc. Then you will treat it with eggs. So you will put eggs on top of it so that the larva can hatch into that substrate and consume it. After some period of time, you will then harvest the larva. A subset of those larvae will be kept out as pupa to be allowed to turn into adults. And those adults will lay more eggs that you can use to treat more substrate, and so on and so forth. So this is what that looks like at scale. Each of these blue plastic bins is going to be bringing about 10,000 black soldier fly larvae. So you can see they're stacked about 5 high by 4 across by 2 deep on these pallets, and those pallets stretch all the way down this greenhouse at JM Green in China. This is what adult rearing facilities look like. So you can see each of those little black dots on these mesh mating cages is going to be an adult black soldier fly. Those lights are some special black soldier fly lights that are meant to work well with their photoreceptors. But you can see they're not provided with food or water in this particular example. So when we're reviewing welfare concerns in rearing and slaughter, we're talking about this part of the process, right? Everything that happens to the larva from egg hatch to slaughter, and everything that happens to the pupa and the adults as they're going through the eclosion and breeding part of the cycle. So in this publication that you can read at the link at the bottom of the screen, and which will appear again later in the presentation, we identify a couple of major welfare concerns for black soldier fly currently being farmed in the industry, some at the adult life stage, some at the larval life stage. I'm going to focus on 2 of these that I have continued to work on empirically for the remainder of the talk today to try and demonstrate the ways that I think we can make progress in improving conditions for these animals once we gather further evidence on their biology and their needs. So the first of these is going to be the fact that adult black soldier flies, like I mentioned to you, are not provided with food and sometimes also not provided with water in industry rearing conditions, which is obviously a violation of a freedom from hunger if that animal is capable of eating. So there's been a misconception in the industry for a long time that black soldier fly adults are actually incapable of eating. And this, I know, sounds insane from a vertebrate physiology perspective, but it is not totally insane. Adult silkworm moths actually have lost the functionality of their mouth parts and do not eat at the adult life stage. So there's some precedent in some insect species that have been farmed for a long time. They don't eat at the adult life stage. The problem is just that this isn't actually true of this species. This species does eat at the adult life stage. So that right there is their functional mouth part. It links up to a functional digestive system. If you provide them with food, they gravitate towards it and consume it, and it dramatically increases their longevity. So here what you're seeing is if you provide just water, no water, or sugar and water to males and females, it a little over doubles the longevity of females, provide them with sugar water, and it almost quadruples the longevity of males, provide them with sugar water. Now, one thing I'll note about this is that we actually don't think most insects just eat sugar water. Like most animals, they require proteins, fats, and carbohydrates. So this is not necessarily actually the best diet for them to be consuming, but it is showing that if you provide them with some kind of food, it has dramatic benefits on their survival rate. So one thing I'm doing with my collaborator, Edward Waddell, and we're being funded by an EA Animal Welfare Fund grant to do this work, is we're looking at preferences of adult black soldier flies for different carbohydrates and proteins. Then we're trying to kind of create optimal diets for these animals, first based on their preferences, and then looking at the impact of that on longevity, reproduction, and behavior. So what you're seeing here is some data from our first preference test, just looking at 5 and 10% concentration solutions of different carbohydrates. So we looked at honey, dextrose, and molasses, and found that there was a strong preference for molasses generally and 5% molasses specifically. We also looked at a bunch of different protein sources that are commonly eaten by other kinds of flies. So we looked at agar, casein, and yeast, and found a very strong preference for yeast, which I find, really interesting just based on some of the other fly biology that we know about from other species. So we took those 2 pieces of preference data. Then we went through and said, which of the following protein to carbohydrate ratios do they seem to prefer? So we found kind of the same, an increase in preference of a 1:4 to 1:8 protein to carbohydrate ratio, no difference between them. So this is mixed sex conditions, males and females, 50 of each. One thing that we know from the biology of other flies that people have done the same kind of nutritional physiology work is that males tend to prefer a higher carbohydrate load and females tend to prefer a higher protein load because the demands of laying eggs and producing eggs for females requires more protein. So it's possible that this split between 1:4 and 1:8 is actually just a sex-specific split between what females prefer and what males prefer in this condition. That's probably something worth further investigation as well. So this is just to demonstrate that, we can make progress on some of these issues, that it's not just feeding them sugar water. We need to be considering their full dietary needs. And there is some data that I'm hoping to share with producers at some point soon that this can actually have reproductive benefits for the colony as well. I do want to note that one thing that producers I've talked to about these results are concerned about is that increases in longevity aren't actually necessarily good for their methodology for breeding these animals. So the females will lay these really large clutches of eggs very early in their lifetime, and then they become less fecund over time, just like with vertebrate animals. And obviously, they do not want to keep them alive in that less fit period of their life. So in some ways, it's convenient that they tend to starve to death within 8 to 12 days because it means your population is constantly just the youngest, most fertile flies mating. So if this is something that we're going to start talking to producers about doing as like a strategy for improving insect welfare on their farms, providing food for their adults, we also need to find strategies to convince them that this is not going to devastate their colony cycle's efficiency by causing lots and lots of old, reproductively unviable flies to be gumming up the works. So we need to come up with strategies for helping them manage their populations differently so that they can provide this welfare improvement for their animals. From the larval perspective, probably the biggest concern, or I guess I'll say not necessarily the biggest concern, but the most immediately obvious concern is non-instantaneous slaughter without anesthesia or stunning being provided. So these are the methods that are typically used for slaughtering insects broadly, not just black soldier flies, also yellow mealworms and crickets. Microwaving is very common, like what you're seeing in this picture here, because of what it does to product quality characteristics. Baking in a convection oven is also used. Other methods are things like grinding, boiling, freezing in liquid nitrogen, freezing in air, or asphyxiation. What we did in our publication was just initially try to review these and put them into two categories of more likely to be humane or less likely to be humane in their particular standard operating procedures. So we know insects are very responsive to aversive heat. So we can expect that things like microwaving, baking in convection ovens, and boiling could be quite painful. The question is whether they are also instantaneous or non-instantaneous. There's some standard operating procedures. Boiling can be instantaneous for insects. There are no standard operating procedures under which microwaving or convection ovens provide an instant death to an insect. You can also imagine situations in which liquid nitrogen could be used instantaneously, grinding could be used instantaneously. Freezing in air is unlikely to be instantaneous, but we're uncertain about what the experience of the insect is like as it's being frozen right now. And the same thing is true for asphyxiation. And so there's some big uncertainties. So now I want to give you an example of some work that I am publishing right now in animal welfare. It just got accepted with a team of people that you can see listed on the bottom of the slide. And this work was conducted in Jeff Tomberlin's lab, who is another co-PI of the site. He's at the Texas A&M site for the NSF Center for Environmental Sustainability through Insect Farming, and he's a black soldier fly expert. So we went through and tried to develop a standard operating procedure that would figure out how that would impact time to death in terms of different variables that matter to producers like larval body size, loading rate, or particle size, plate hole diameter. So this is the example meat grinder that we used for this study. So you can see that it's got kind of a tube that the larvae get fed through, then there's a blade that they will hit, and then right after that blade is the particle size plate that they get pushed through. And those holes can differ in diameter from 12 millimeters down to 2.55 millimeters. So what we found when we did this work was first that larvae can have two different experiences using this kind of machine. They can actually make it through the particle size plate, in which case they become kind of a mush, like what you're seeing over here on the left, or they can get stuck behind the particle size plate. And this is the typical experience of the larvae that get loaded last. And that's because there's nothing behind them to push them through the particle size plate. So they get stuck at the blade instead. So you can see that happening in this picture. So what we found is that as we decreased the size of the particle size plate hole diameter from the 12 millimeter down to the 2.55 millimeter, more larvae got stuck at the blade because more force was required to push them through the particle size plate. Also, large larvae are more likely to get stuck compared to small larvae because, again, probably more force is required to get them through the particle size plate. So there's a difference in the number of larvae having a particular experience based on these two variables. The problem with this is that if you get stuck at the blade, you only have a 3% likelihood of instantaneous death. So you are extremely unlikely to have a humane experience if you do not make it through the particle size plate. And this is just some examples of that for insects that either made it through the particle size plate and got injured but don't die immediately, or those that got cut in particular body segments, the blade, and did not make it through immediately and die immediately. So you can see that they can get injured and remain alive. In fact, we quantified for different larvae of different body sizes how likely they were to get cut in particular body segments and instantaneously die. So these colored bars are just talking about different body segments from segment 1 to segment 12, because they have 12 body segments total. And you can see that it's only kind of in the middle that you're seeing any likelihood of instantaneous death. So that's like segments 3, 4, and 5 for any body size larva, from the smallest 7-day-old ones up to the largest 23 to 24-day-old ones. And even if you wait 90 seconds, still relatively few of them are dead by 90 seconds if you get cut in these particular body segments. So if they're getting injured, but not completely ground up and disassociated, if they're just receiving a single cut, they're very unlikely to die instantly, even making it through the particle size plate. So we then used this data to figure out what the likelihood of instantaneous death was using these different hole diameters. It turns out body size did not impact this, just hole diameter impacted this for all the larvae in a run. So what we found is that if you are going through the largest hole diameter, you get a 54% likelihood of instantaneous death. If you go down to the smallest 2.55 millimeter hole diameter, you get all the way up to an 84% likelihood of instantaneous death. So first thing we can clearly recommend for producers is smaller plate sizes are better. Smaller hole diameters for your plates are much better from a humane perspective. But remember that stuck at the blade population I told you about? They're actually dragging down the likelihood of instantaneous death for that small particle size plate. So if you make it through the small particle size plate, you have a 99% likelihood of having an instantaneous death. It's the only 3% of them that are dying instantly at the blade that are dragging that number down to 84%. So this gives us another recommendation we can make to producers. They need to do something about that population at the end of their loading period that's getting stuck at the blade. They need to run some kind of filler through at the end of a batch that's going to push those larvae through and make sure they also have 99% likelihood of instantaneous death. So these are the kinds of recommendations we can start to make to producers to increase the likelihood that their standard operating procedures can be more instantaneous and more humane for larvae. Now, again, as a realist, I want to note that this particular method might actually have some challenges with adoption. One of them has to do with oxidation rates. So as you can see from the material in this picture on the left, the fat in those larvae is already getting very dark, and that's because it's oxidizing due to the amount of contact it has with the air. So oxidation is one of the major problems for this industry, where lipid oxidation leads to very low product quality and bad shelf storage. So they are very wary of methods that increase oxidation rates. Grinding is probably one of the methods that increases it the most because it ruptures the body and leaves the lipids totally exposed to air. This is why they tend towards using things like microwaving, convection oven baking, things like that. So further work is needed on those methods, and potentially on developing stunning technology that can be used prior to the employment of those methods to make them more humane. But I will say that one area where I see potential promise for this method is actually as a method for depopulation, where product quality is totally irrelevant. So we often have cases where insects are failing to thrive and producers don't know why. And right now, some producers anecdotally report to me that they might just toss the insects into a garbage bag and throw them out. This is probably not likely to be a super humane experience for those larvae if the reason they're failing to thrive is because they're suffering from a disease or something like that. And so we might want some kind of depopulation method that is a little bit more humane and intentional. And this might be good for that as a process. So if that were the case, we do need to test it on even smaller body size larvae than what we tested, because we only tested slaughter weight relevant larvae. And we need to test it with the substrate still mixed in, because they're unlikely to want to separate the animals from the substrate material before running them through the grinder. But assuming that we can do both of those things, this might be a more humane depopulation method than anything we currently have. And so I've talked about a couple of things for the black soldier fly as case studies. There are obviously many other welfare concerns I didn't get to go through today for that species. Many of these are in common across all the species that I focus on: crickets, mealworms, and black soldier flies. So all of them experience inhumane slaughter, all of them are photophobic, and yet can be subjected to light during handling. But we also, just like with cows and chickens and pigs, we have different welfare concerns for crickets, mealworms, and black soldier flies. We have different welfare concerns emerging based on their biology, their physiology, and the conditions in which they're reared. So you can read more about all of the different welfare concerns that manifest for these species in these 3 publications, which if you look up my research profile, you'll be able to find them. They're in Journal of Insects as Food and Feed, the first 2, and then the last one is being resubmitted post-revisions right now. And I want to thank Rethink Priorities, which funded all 3 of these review projects. And also, because we're vets here in the room today, I wanted to bring up a couple of things that I think are especially relevant to your population and your interests. One thing that I think about that I lose some sleep over sometimes is biosecurity concerns in this industry as it develops. We know insect escapes from facilities are common, even. They have a lot of historical precedent. So like Africanized honeybees escaped from greenhouses. Disease-carrying bumblebees that are invasive in North America escaped from greenhouses. Spongy moths escaped from this guy's facility in his backyard in the 19th century. So these have caused a ton of problems, from disease spread to wild populations of insects, huge consequences for our ecosystems where they have destroyed a bunch of potential species or really radically altered various ecosystems. It can have significant economic damages depending on the industry. And in some cases, like the Africanized honeybee, it can even lead to human deaths. I don't think that's likely for many of these species, but I think the other things are a little bit more likely, things like economic damages or the spread of disease. Right now, again, producers have no assistance for managing biosecurity or disease risks, the risk of release of GMO or selective breeding lines that they're making. And producers, like I mentioned, they may not be treating those failure to thrive populations before they're releasing them into the environment. If the reason that that population fails to thrive is because of a disease, and you're just putting them in a trash bag and putting the trash bag outside, if some other wild animal comes and rips open that trash bag, you can have insects come to that trash and start to spread that disease throughout the environment. So there might be some biosecurity concerns that we could take more seriously in this industry to help producers mitigate that risk and mitigate the risk to their surrounding ecosystems. Another problem is that producers often swap populations with each other as a way of maintaining genetic diversity among their populations, but this has been known to facilitate disease spread among facilities, so there might be room for education or work there about appropriate quarantine periods and protocols. And the last thing I'll note is that we also know that at least for crickets, some of their iridoviruses actually can spread to lizards and other exotic pets. So there's something to be thought about there too in terms of the exotic pet feed industry and the use of these animals there. So kind of in conclusion, farmed insects, there's a lot of them. There may be more evidence for pain, suffering, sentience than you initially thought, even if we certainly are far from proving that they can feel pain. There are many serious welfare concerns that are different for these different species. Novel welfare concerns will continue to emerge as this industry continues to develop. And so there's a significant need for collaboration to be able to do our best by these new livestock animals. Last couple of things I'll note, if you're interested more in bees, you might check out the work of the Honeybee Veterinary Consortium and the Veterinary Invertebrate Society, which are two groups that have focused on the managed honeybee problems and potential interventions. In particular, in the United States, they focused on the fact that there are requirements now that if you're going to use antibiotics with your bees to treat disease, you need to get a veterinarian's prescription. So that's part of what the Honeybee Veterinary Consortium was founded to address, and their work is very interesting. But that's another population of farmed insects or managed insects that I don't work on much, but people are often interested in. And many vets work on the research context. So if you're somebody who works with laboratory animals, we use many billions of insects each year in research. There are currently no ethics guidelines or reporting requirements anywhere in the world for insect welfare. We have created, through the Insect Welfare Research Society, the very first guidelines on how to protect and promote insect welfare in research. But there's a lot we don't know. So if you read those guidelines, you'll see that there's a lot we don't know about humane anesthetics and analgesics, humane euthanasia protocols. And so that's something that the community of researchers that work with insects also could really use help on, and that veterinarians are probably best suited to help address. So with that, I will thank Rethink Priorities and EA Animal Welfare Fund that have funded various empirical projects I've talked about today, and I would be really excited to take your questions.
Awesome. Thank you so much. That was really, really interesting. We do have a few questions in the chat. Heidi asks, how might sociality play a role in sentience and or pain and associated welfare implications? Might insects with a social brain experience pain differently? Have there been any studies investigating differences between highly social species, e.g. ant colonies, and less social insect species?
Yeah, so a couple of things come to mind there. I mean, on the nociceptive side, so not the pain experience side, but just like what's the nociceptive neurobiology like? We see a lot of similarities in the nociceptive neurobiology of the solitary and the social species. So from that perspective, my immediate response would be, well, they have relatively similar TRP channels and they have the same integrated brain regions. So not so sure it would be different. On the other hand, we do see some very significant differences in the organization and the kind of the size and structuring of certain brain regions of social versus non-social species. So for example, our bees and like our social bees, they're known to have very, very dense mushroom bodies, which are the center for learning and memory for insects. So they have a lot of cells in that part of their brain, many, many, many more than you would expect if they were a fly, for example. And you might imagine that along with those additional cells in that region are some additional capacities that might lead to more complex experiences, more complicated kinds of learning, things like that. So I would say the jury is very out on whether the social species and the solitary species experience pain differently, or perhaps have novel dimensions to their pain experience where, okay, maybe their base individual experience of pain is the same, but then you can have a layered effect where, like maybe the social species, um, maybe get some kind of magnified effect from the pain of their group mates, for example, whereas solitary individuals would not. We do not know anything about that. It's all speculation, wild speculation on my part.
Yeah, that makes sense. And Andre asks, do you know what the industry terms thermal shocking to mean in the slaughter sense?
That's a good question. I don't. I mean, I think, is that used for insects, or is that typically used for like shrimp in aquaculture?
Andre, feel free to unmute if you're around. I don't know if I've seen that personally. If he's not around and can't clarify, do you have any
So
further
there are,
thoughts? You heard of?
you have some further, I can again speculate wildly on what
Yeah,
I think that term might mean. Oh yeah. Hey, Andre.
yeah, just a large black soldier fly farm is claiming that the method that they use that they will use will be thermal shocking, and I'm trying to figure out what that means.
Hmm. So there's two ways. Thermal shocking generally just means subjecting the organism to a very different temperature very rapidly. But I think it can be applied in the cold sense or in the heat sense. Mhm. My guess is that they likely mean the heat sense. I haven't seen that many people. The way you would do thermal shocking on the cold sense at scale would be something like liquid nitrogen, and it's just expensive. So I haven't seen many people willing to do that. Whereas on the heat side of things, you're just talking about boiling water or steam or something like that. And so people are much more likely to do something like that. So my guess is that by thermal shocking, what they're talking about is either boiling or blanching. I guess what I would say there is that is on the list of like more humane, but it's very standard operating procedure dependent. And what I mean by that is if you take a single larva, drop it in a pot of boiling water, it will reach its critical thermal maxima, its lethal temperature, within a second. But if you take 200 larvae and drop them in a small pot of boiling water, they will not all reach their critical thermal maxima instantaneously. So it's very dependent on how much water, how many larvae, the shape of the container, the temperature of the water, the temperature of the larva, the thermal kinematics of the system. And there is no guidance right now for producers on what standard operating procedures actually work. So my concern there is that producers might try to save energy by, say, reducing the temperature of the water or something like that, or by adding more, save time by adding more larvae to the pot than, they would otherwise, and that these kinds of things would cause it to be non-instantaneous and thus quite painful, potentially, for the larva.
Interesting. Yeah, we're already seeing that with chickens and bird flu and using ventilation shutdown to kill them. We're seeing producers want to lower the heat to save money, as the USDA is now considering just giving flat payments to these producers. They're like, well, how can I get the most out of this? So I'll just lower the heat so it takes longer to kill the birds. So yeah. Christina says one would hope that they would use something like clove oil on the disposed insects so they don't get released. I don't understand really, but do you know what she means there?
So clove oil, we don't know whether or not and at what concentrations. So I guess I would say clove oil anecdotally. I talked to one person who has worked on it in a research lab, but she hasn't published on it yet. And she found that the clove oil takes a very, very, very long time for bumblebees to take effect. So my view would be that something like the grinding method would be potentially better. And then following that up with some kind of disposal, like incineration or something like that, to make sure that any diseases that are in that material also go away. Because it's not even just the insects getting released, potentially, the diseased insects being released. It's also the fact that they are diseased material. Like even if they die of that disease, all of them, if like a badger comes over, rips open the trash bag, and then wild flies come to that trash to like eat the food that's there, they will pick up the disease from that food. And so it's important that that material be dealt with in a safe and secure way. We would not, I actually don't know that much about vertebrate livestock, so I apologize if what I say is about to be incorrect, but I assume you are not supposed to just like take the birds that are diseased after ventilation shutdown and just like kind of put them outside. You're not worried about them You're not supposed to. I said you're not supposed to. Because other birds could come along, visit that site, and then pick up the disease and take it with them and have devastating ecological consequences.
Mhm. Yeah, you would think, but yet they do. It puts some of these birds just out there and covered with a little bit of soil. So yeah, it just, you see what happens when you don't clearly lay out and also enforce the rules that you should be making to prevent biosecurity risks. Andre asks, is there any evidence to suggest that overcrowding might be a welfare issue for crickets, mealworms, BSF larvae? Is there a threshold of crowdedness where they will avoid conspecifics or try to create space?
Yeah. So definitely for crickets for some species. So that's what I was talking about with the self-thinning mechanisms. So you have two types. You have the literally we are going to eat each other until there are fewer of us around because we're so stressed by how many of us are here. So that's one mechanism for crickets. The other mechanism, and you can read the actual densities for each species in the publication that I have online on OSF preprints. I actually go through and list like the specific, as much as we know about, because honestly, we need more information, but as much as we know about what densities seem to trigger these problems in crickets of different species. The other mechanism that they have is that dispersal mechanism I mentioned, where some species have those two morph types. So they have long-winged morphs and short-winged morphs. And again, any individual can end up with long wings or short wings, depending on the density when they're being reared. So they develop longer wings if they are trying to get away. And that suggests to us this is too high of a density. You need to lower the density because they're trying to disperse. They cannot disperse on farms. So for crickets, absolutely. For mealworms and black soldier flies, the case is a lot more complicated. We don't have any data that suggests there is a density threshold that causes behavioral changes in either of those, well, in black soldier fly larvae, that they want to like get away, for example. What I can tell you is that if you rear too many of them in too small of a space, you run the risk of them overheating because they actually do, in aggregate, produce a pretty large amount of metabolic heat. So then you get the center of your aggregation getting very, very, very potentially lethally hot for the individuals in it. And so then they are trying to escape, but it's not actually clear they're trying to escape because it's crowded. They might just be trying to escape because it's really hot. And so indirect effects for those two species, the direct effects are much less certain. The other thing we know from mealworms is that if they are in too crowded of an area, it can delay their pupation. So again, if you imagine the pupal stage is one of the most vulnerable stages for that species because they are much less mobile. And so they can be eaten by their conspecifics much more often. They look for pupation sites that are further away from conspecifics. So they are most likely to avoid being consumed by their conspecifics while they're trying to pupate. If they're being reared at really high densities and they're constantly getting touched by things, they won't pupate on time.
Interesting. And Amy brings up a good point, which is why dog and cat veterinarians should care about insect welfare and why we should also care about any farmed animal welfare, because sometimes our patients eat the patients of those who do this sort of work. So do you know what mechanism and how is the quality of life for the dog? I guess there's insects used in the dog food company, Jiminy. Are you familiar with this brand and what they do?
Yeah, so I'm not familiar with this brand and what they do. And I'm not familiar with which producer supplies them off the top of my head either. So unfortunately, I can't comment on any of their practices.
Okay. Yeah, I'll be interesting to research more into that and what these various companies do. I personally am wondering if you have looked at methods of parasite control for animals, if this has come up at all, flea, tick, heartworm prevention for dogs and cats and other species, and intestinal parasite deworming. I mean, this is a self-defense issue for animals. And so many of us believe removing parasites is deemed ethical because of this. But are there methods of parasite control for animals that are higher welfare for the insects than others?
It's a great question that I do not have. I've not been asked this before, actually.
Oh,
I've been
wow.
asked about like pesticides and stuff, but I haven't been asked about parasite control yet. One thing to keep in mind is that some of your parasites are not actually, that you're dealing with are not actually insects. Obviously, your fleas and ticks are not technically insects, but they're arthropods, at least.
Oh.
Like a lot of your intestinal worms are not going to be arthropods and stuff like that. And so we might have different levels of certainty about their sentience that I can't comment on because I'm not an expert in those groups. So then once we look at the insect or arthropod question, again, my guess would be that you're looking for things that are likely to be faster. You know, I mean, people will sometimes ask me like, oh, if I come across an insect in my house that I'm unwilling to allow to be in my house, and I, don't think I can move it outside, what should I do with it? The answer is you should crush it as quickly as possible, right? If you are committed to killing the insect, you should kill it as fast as you can, because we know so little about what causes them pain of what degrees right now that it's very hard for me to adjudicate, oh, put them in the freezer versus boil them versus, shred them. But instant means instant, right? So if you can instantly kill them, that's the best way to go. I don't know as much about how these different parasite control agents work, so it's hard for me to know which ones might be better or worse. My guess would be like, I know I've given a lot of flea baths to kittens myself. Probably not the best, actually, from the flea's perspective. Probably not fast.
Interesting. This is something to think about, like as dog and cat vets. I think we get a lot of continuing education from these various companies that do insect and parasite control and things like that. And maybe we should, when they come and do a CE talk, that should be one of our questions as dog and cat vets ask is, can you tell us about the welfare of those insects, how they are killed, and encourage them maybe to do more research in this area too. So
yeah, and it's not impossible to do that. I think do often get a lot of very skeptical responses from people, usually on the malaria question. That's where it comes up. People are like, oh, so like what, we stopped treating, we stopped trying to kill mosquitoes that carry malaria now. And I'm like, I see, we're genuinely interested in this topic. And my response is typically something like, well, actually, I went to this special interest group meeting on ethics and the treatment of insects. And we had a company that produces anti-malarial products, anti-mosquito products for malaria circumstances come to our the welfare and ethics meeting. And they presented on a higher welfare insecticide that they're developing for mosquitoes that carry malaria. And it's much more targeted. It affects many fewer non-target insects. So that's one way that they can reduce the impacts, is by making it a more specific type of pesticide, and a faster acting one. And so, yeah, you actually can take these topics seriously, even for mosquitoes that carry malaria, and have conservation benefits and ecosystem benefits as well as welfare benefits for the animals.
yeah, and I just think if we start studying welfare more, we will come across things that will ultimately benefit our own welfare and the welfare of everyone. Like when we start looking at these things from this perspective, I think we'll open a lot of doors to improving the lives of everyone. And Amy asks, if you find an injured insect, do you kill it to end suffering or leave it just in case it can recover? Always a debate for me. And sounds like they can survive a lot of injury based on this talk.
Yeah, so I think it depends on what the injury is. There are some injuries, like if you come across, for example, a bee and it has an injured wing, that injury actually might not be causing that bee any pain, in that the wings are not likely to have a lot of nociceptive ion channels in them. However, that bee can't fly anymore. And the likelihood that that bee is going to starve to death is extremely high, or freeze to death because it can't get home to its colony, is very, very high. So that might be a case where you, by killing the insect very, very quickly, are actually benefiting its life overall. But if you came across that same bee and it just was missing a leg, like insects can do a lot with missing legs. Now, I will say sometimes they develop diseases from that, right? We see that any kind of particular injury opens the door to pathogen infiltration. So there is the possibility that that insect will develop a disease later on that will lead it to die and suffer. But at the very least, I can't, that one's up in the air for me. The wing injury, not up in the air. That insect's just waiting to die. So I think there's some context dependency to it like that, where you look at them and you say, is this an insect that's in a context where it's likely to suffer as a result of this particular injury? Or do I think there's enough of a probability it could survive and be fine that I'm happy to move on?
Yeah, interesting. Yeah, I recently came across this. I was in Florida and this giant bug, I have no idea what they were, was in my Airbnb, and on their back. I think some sort of cockroach, giant cockroach,
Probably.
picked them up,
Never
put
getting
them outside, and flipped them over. And they were kind of twitching, but not really moving. A few hours later, they were back on their back. And I'm like, your chances of survival out there in the wild are not good.
Not high.
Might
Yeah,
be
probably a cockroach that came across a neurotoxic pesticide that somebody had sprayed indoors to try and get rid of the cockroaches.
possible, something like that. But this was really interesting. Monica, is it a question? Really informative. I learned a lot, and I'm also simultaneously saddened by all we are inflicting on insects now. Thank you for the work you do. Yes, thank you for this work. And this is so fascinating. And I hope that there's more people who are inspired to join your field and help all of these insects, because there are so many of them, and we all interact with them on a daily basis, and we know so little about them. So this was really
Yeah. I do want to thank everybody for coming and making the time. I know it's a very strange, novel topic that we're delving into today, but I really appreciate everybody, coming, paying attention, listening. Also, if you want to stay informed on this topic, the Insect Welfare Research Society, everybody can sign up for our listserv, and we send out newsletters once a month with additional research and information on the topic. So you can stay informed about, studies into euthanasia, studies into slaughter, things like that as they come out.
awesome. Great work. And thank you all for coming, and I'll see y'all next time. Bye, guys.
Thanks.